Λοιπόν, με ιδιαίτερη χαρά να σας γνωρίσω τον τελευταίο ομιλητή για το δεύτερο Πανελλήνιο Συνέδριο Εξατομικευμένης Ιατρικής, last but not least, έτσι όπως λέμε. Είναι ο αγαπητός μου Νίκος Κατσάνης. Ο κύριος Κατσάνης υπηρέτησε ως καθηγητής παιδιατρικής και γενετικής ανθρώπου στο Feinberg School of Medicine στο Northwestern University στο Σικάγο και είναι σήμερα συνιδρυτής και Chief Scientific Officer της εταιρείας βιοτεχνολογίας Galatea Biotechnology στην Φλόριντα και θα μας μιλήσει σήμερα για ένα πολύ όμορφο θέμα, 1+1=0. Νίκο, σε ευχαριστούμε πάρα πολύ. Μας τιμά η παρουσία σου και we're looking forward to your talk.
Ευχαριστώ, Γιώργο μου, για την πρόσκληση και σένα και την Οργανωτική Επιτροπή. Και πρέπει να ζητήσω συγγνώμη σε όλους, γιατί δεν μπορώ να σας δώσω την ομιλία στα ελληνικά που κλείεται, δεν τα ξέρω. Αλλά θέλω πραγματικά να σας πω λίγο σήμερα 1+1. Με ρωτήσανε ένα-δυο φίλοι τι είναι αυτό, λέει, 1+1=0. 1+1=0, ελπίζω να μπορέσουμε να συμφωνήσουμε στο τέλος της ομιλίας ότι η ανάλυση του genome είναι πολύ πιο περίπλοκη από ό,τι πιστεύαμε. Λοιπόν, ας ξεκινήσουμε. I'm sorry, I'm going to switch to English, but I will try to go at a reasonable pace. I not speak Greek pace. So, in ancient times, as in before the year 2001, if we think about the way we interpret the genome, and especially with regard to drug discovery, which is the area that I'm going to focus my talk today. So I should preface and say, it's all fine and good to sequence genomes and find new genes, and look, here's another one. But I think we're reaching the point now where we're beginning to answer the question, what we're going to do regarding this information, how we're going to transmit this information to individuals, and how we're going to improve individuals' health care. So back then, if you thought about how we discovered drugs, there's about 600 or 700 drugs for rare and orphan diseases, another 200 or 300 drugs for more common disorders, and that's it. That's the grand sum of everything we have accomplished, which is awesome. If any of you have time, think about science and medicine in the year 1700. So in a short period of time, we've accomplished a lot, but we still have a long way to go. So if you think about drugs, back in the day, we used to do phenotypic screens on core cell physiology. So what you did is you took some sort of cell line that sort of have a particular property, it glowed green, or it moved across a dish, and you threw a bunch of compounds on it, and then you said, because some small number of compounds did something good, and then you did more and more, you failed, you spent millions of dollars, and then you tried again. Or you did targeted screen, a kinase that is mutated in some cancer, I'm going to devise a scaffold around it. If I think about where we are today, I really cannot help but be thankful that I'm able to live and work now, because new tools have come online that are really changing the way we'll be dealing with this problem. If you think about this, and I know that this conference here has dealt with this a little bit, and I certainly hope to collaborate with many of you to this part of this effort. We are now, across the world, we're amassing large cohorts with electronic medical records, and each of these cohorts has some level of omics attached to it. You know about FinGen, you know about the UK Biobank, you know about Go Netherlands. I know there's a Greek genome project that's been discussed here. I think that's fantastic. And so on and so forth. There's about 60 countries in the world that are systematically collecting genetic materials from their citizens with associated clinical data and trying to do omics on those individuals. Genomics, proteomics, metabolomics, microbiomes. For those of you who think about this, things have gone very well here, and the old guard might remember that it took 3 to 4 years to make crystals from one protein and then bombard it with X-rays and then go find time on a supercomputer to derive structure. We now have over 45,000 structures. And now we have enough information that given advances in machine learning, the rate at which we're accumulating new protein structures is truly extraordinary. And not only are we finding structures, and I want especially the students in here and the younger people, I want you to encourage to look at some of this stuff, because there's new technologies that not only are we learning about protein structure, but how proteins are moving inside of fluid. I know the books show pictures of cells like a Snoopy cartoon, but don't forget, it's a bag of water. So every protein actually has mobility in a liquid medium. Some other things that are going on, we now have massive and more accurate protein-protein interaction networks. Many of you will know about this, and I would imagine that some of you at some point will or have or will look at first neighbor analysis and look at networks of your favorite genes and proteins. And of course, the other thing we have is machine learning to optimize discovery at a rate that is truly unprecedented. Okay, so here, this is the good news. Here is the not so good news. This is total number of drugs approved per year from 2008 to 2019, whatever it is, 2020. Yes, it is increasing. However, this slide is deceptive, because a lot of the stuff that is increasing is actually, in Greek, alexomanoios και έβαλε τα ρούχα του αλλιώς. Okay, so it's the same drug that has been remarketed under a different name. Oh, you know, I'm going to put a hydroxyl group at the end. It's going to look something very different. Now, no, it's the same drug. Or it's the repurposing of the same drug for a different indication that requires another permission. So, in essence, all the amazing things I told you in the previous slide have not yet really translated into the drug discovery paradigm. The other thing to tell you is the National Institute of Health in the United States has about a $38 billion budget. That's how much they spend, the United States spends. The top 15 pharmaceutical companies together have an R&D budget of $150 billion a year. What the hell are you doing with all this money? Because it's not showing here. R&D costs are increasing, the tools are getting better, but we're not getting better at discovering drugs. The rate of drug discovery is not increasing. The system is broken. We need to rethink these people. Now, unfortunately, I'm at the edge of the old people band. I'm not quite there, but I'm working on that, which means it's up to you lot to solve that problem. And I'll give you a few ideas. We know this from a study done from GlaxoSmithKline in the early 2010s that showed that human genetics remains among the strongest predictors of success. In other words, if you know that you have a target, a point mutation suggesting a particular phenotype or engagement to a small molecule or whatnot, the probability of success at a clinical trial is going to be much, much higher. So then, if we know this, and some companies already know this, and including people you already know. If any of you haven't looked at this, GPCR76, non-such mutations associated with obesity. That actually came out primarily of a cohort of 100,000 people from Mexico City, and it has now represented now $1 billion contract between Amgen and Regeneron. So what I want to really think about is how do we leverage smart genetics to both improve biology and drug discovery? And we really need to start shedding a few of our preconceived notions. Because unfortunately, even though in the medical profession, we would love it to be, you know, the black hats and the white hats, good, bad, this is Tarzan speak. The reality is that everything is, the answer is always depends. And those of you who are dealing with the genomes, especially in the clinical setting, know this much better than I do. So the classical view is that deleterious mutations are deleterious. That is patently false. And I'm going to explain why. It is true that a deleterious mutation is deleterious to protein function. What is not necessarily true is that loss of protein function is deleterious to the phenotype. CCR5 deletion, as you all know, protects against HIV. Interestingly, we now know that COVID-19 has tropism against CCR5. Could it be that we're going to have the same protection effect? I don't know. I'm wondering. PCSK9, the most famous one. Familial nonsense mutations in PCSK9 are associated with low cholesterol levels. We now have two companies that have produced drugs based on this paradigm. So, the fundamental premise that I really want to talk about, and I will talk about with examples, is that loss of function genetic events can actually have therapeutic value in the context of other disease-causing mutations. And that systematic identification of such sites can identify key therapeutic targets. So I'm going to spend the next 2 hours, on three things. I'm going to show you an example of evolutionary genomics. I'm going to talk a little bit about functional screens, and also from gene discovery to new treatments. So, the first example, I really want to think about species and context-specific effects. And I really want to emphasize this, especially among those of you who are dealing with discovery of new point mutations in patients with various disorders. So I will be the first to raise my hand and say, until I was a little bit illuminated by data a few years ago, if I saw a pathogenic mutation, a point mutation, in some gene that I was studying, and I found this mutation in another species, I would go, bah, it's probably another rare polymorphism, right? However, it's not quite as simple. So let me show you an old example. This is a study done many, many years ago, where we showed that mutations in this gene, RPGRIP1L, were associated with very severe ciliopathies. This is a group of diseases I've been working on most of my career. And what was interesting, reviewer 3, it's always reviewer 3, actually said, well, that can't be because we have this mutation here, arginine 937 to leucine, and we had two species in which the leucine is fixed. Now, this disease is neonatal lethal. How can you have a recessive neonatal lethal disorder and one of the pathogenic mutations being fixed in horses, and I don't remember what the other species is anymore? The likely explanation is that this is just a rare polymorphism or a rare variant that is irrelevant to disease. Yes? Yes? Thank you. Except when we functionally tested this allele, so this is a zebrafish embryo, the head, the tail, and this is a very simple assay. Basically, you have your phenotype here, these are your controls, then you suppress the expression of this gene and you get some phenotype, and then you co-inject morpholino back in those days. Now we also do it with CRISPRs. You co-inject it with wild-type human mRNA and you rescue, and then you take the wild-type human mRNA and you introduce the point mutation, and the point mutation fails to rescue. There can only be one interpretation to this result, to this observation, and the interpretation is that this mutation is loss of function. So here we have a paradox. We have a paradox in which we know that loss of function of this gene in humans is not tolerated, and yet we have an allele here that the functional data says loss of function, but the evolutionary data say, nah. I am going to bet you €1 that those of you who study alleles like this have come to these examples all over the time. And most of us, including me, tend to throw them away, disregard them. Too difficult, we don't know. Okay. Why? So this bothered me for many, many years. And then I was talking to Shamil Suneyev. Some of you might know Shamil. He wrote Polyphen. And I said, you know, your program sucks, man. And then we became best friends. And he said to me, yeah, no, it doesn't deal with evolutionary constraint as well as it could. So anyway, Daniel Jordan, who was a graduate student in Shamil's group, said to me, you know what? Let's just figure this out. Let's figure this out if your RPGRIP1L is some weird thing or if it's a common theme. And what Daniel did was cool. So what he did is he took everything in HumeVar and ClinVar. These are two databases that some of you might have used, and he took all alleles. Now, I know there's rubbish in each of those. Every group of data has false positives in it, but the number of alleles here, and he aligned them with 100 vertebrates, and he asked the question, how many alleles found as disease-causing HUMVAR or ClinVar are found in at least one vertebrate? And the answer is shocking. The answer is anywhere from 3 to 12%. The median is about 7%. So my statement to you is about 7% of all the mutations reported to be pathogenic in a human being are found fixed in homozygosity in at least one model organism that we now know of. Now, this study is already almost 6 years old. I bet you if we did this now, we'd find even more. And I will bet you that if we were, forgive me, honest and actually reported everything we find, as opposed to the things we think are pathogenic, then these numbers will be much higher. How is this possible? So I came across this study. So it turns out that those of us with the human genetics, don't know everything. There was this beautiful paper that I came across. Shamil pointed this to me actually. And this is the concept of compensatory deviation. So how does this work? Imagine that you have this protein here, and then you get a mutation. And something bad happens to the protein. It doesn't fold very well, it gets degraded. But you could have a second mutation that actually corrects, compensates the physiological structure or the physiological defect of this mutation. And therefore you have compensation, which means that if you have a species that happens to carry the second allele, the genome of that species will become permissive to the presence of the first mutation. Yes? Okay. Now, like my old prof, Jim Lupsky, had a sign outside his office. It said, without data, you're just another dude with an opinion. So here's some data. Here is one of the many families that we studied over the years. This little girl, we recruited her when she was 18 months old, and she had a clinical file about this thick because she was one of those genetic odyssey kids that some of you might have had experience with. She had developmental delay, microcephaly, various feeding issues, and so on and so forth, a whole cohort of clinical testing. And it turns out that when we did quad-based whole exome sequencing, we found at this point the obligatory mutations in titin, because everybody has mutations in titin, and 2 de novo mutations, one of them in NOS2, and one of them the gene that we hadn't seen before in humans, mutated humans, called BDG2. All we knew that it was some sort of G1S cell cycle transition. Okay. All right, so this is where, if Stefan Frangakis were here, we'd be like, go, because this was like 3 years of his work that it will be now one slide. That's the way it goes. So here's the locus BDG2, the nucleotide change, the amino acid change, the allele frequency. It's a valent to methionine at position 141, and polyphen, SIFT, mutation taster. I can go on. Everybody reports the allele is benign. However, this experiment that was done with Stefan and also Maria Cusi in the lab showed that the allele was pathogenic. So what you look at here is one of the experiments. And in this particular experiment, we are measuring how many cells are born in the head. So this is a mitotic marker. And you can actually see there's a significant difference. You can quantify this using automated imaging. No, I did not get a graduate student to count these dots by eye. That would be cruel and unusual punishment. Okay. But these are the absolute numbers. So this is the number of wild type, suppression with wild-type human mRNA, and suppression with an mRNA encoding methionine at position 141, loss of function. All right, so what did we do next? We asked the question, why is this allele predicted to be benign? And the answer is because the majority of the animal kingdom has methionine, not valine. In fact, methionine is the ancestor allele, and valine is the derivative allele in the primate lineage. So yes, I'm telling you there's a little girl who has a very severe disease because she has a heterozygous de novo mutation valine methionine, and that I have 38 species that are homozygous for methionine and are fine. Your dog has methionine. Your dog does not have microcephaly, seizures, and feeding difficulties. Okay, so does your cat and your horse and whatever else you have. Don't know about chickens. So the experiment with that was very easy. I'm showing you a sequence from a different gene, but the concept is the same. And I will tell you that there was no Illumina instrument within a mile of this experiment. This was a few people sitting down and having a decent conversation and using the best computer you have around, our brains and our interaction with one another to learn and to teach each other how to do an experiment. The experiment was very simple. Here's the sequence. In this particular sequence, we have a mutation of A to human disease. We align these with a species that have the disease allele, and we ask the question, what other point mutations are present in those species but are absent from the human sequence? Those alleles become candidate compensatory alleles. Does that make sense to everybody? Yes? Great. And then, so what is the experimental test? The experimental test is we take the human sequence, we introduce the point mutation, and when we start introducing each of the other point mutations, and we ask the question, are we converting this to wild type? And of course, that's exactly what happened, and I'm giving you the shot in one bang. This is the phenotype I showed you earlier. So this is head size, essentially. This is neurogenesis. So look at this thing. Control, suppression, suppression with rescue, and then suppression with this valine 141 methionine, and then on top of this guy, we'll start putting other mutations. And it turns out that there's two mutations, arginine 80 lysine here, and leucine to valine. I mean, who gets out of bed for a leucine to valine change? It's almost the same. I mean, they're very, very similar amino acids. And yet it is true that if you introduce this in the context of the valine mutation, you get complete rescue. And I can tell you that just this change, or this change, just one amino acid change, explained 32 of the 38 species in which we found the change. How is this? Well, this is where a little bit of 3D structure helps. So this is the actual solved 3D structure of BDG2, human wild type. So you have the head of the protein, this unstructured region here, and then you have this tail, and it turns out that this tail gets ubiquitinated and regulates the degradation of the protein. It turns out that if you have valine to methionine, this tail will flip out like this, outside. But if you have valine to methionine, and this is Matti Ketsio's work, if you have valine to methionine in the presence of either R80K or leucine 128 valine, the kink in the C-terminal tail of the protein comes back in and gets restored. So the idea is we're restoring the half-life of the protein, which is the mechanism by which we get a rescue. Now, great. Is this one example? How does this work? What is the big deal? I think it's an enormous deal. I think that we're missing a diagnostic yield from known genes because we are not examining some mutations that are found in other species. I'm not suggesting that if a mutation is present in other species, it will be necessarily going through these exotic phenomena, but I want to encourage everybody to at least consider this, and I think this merits discussion. The second thing is, we need to start conditioning genomic data analysis. The more genomes we do, the more reductionistic we have gotten about their analysis. The one allele, the one G was hit, the one EQTL, this and the other. It is interesting, is it not, that even though we're saturating the exomes and the genomes right now, the diagnostic yield is not punching above 50% for most things. Where is the other half? We keep discovering more and more genes, and yet. And of course, we have insider protein function and targeting mechanism. Here is where things get really, really scary for me. From literature, so as we know, nobody motivates researchers to publish negative data, which means that you made a knock-in mouse that didn't have a phenotype, you don't get to publish that. From research, it turns out that 20 to 50% of mouse knock-in models do not mimic the human or the knockout. So you take a pediatric enzyme deficiency, you knock out your favorite gene that causes a disease, recessive, you get beautiful phenotype with the enzymology as you expect. You take a point mutation found in human missense and you knock it in the mouse, 20 to 50% of the time that doesn't match, or it is milder, or has later presentation. Why is that? Because humans are not mice, I hear people say. Well, what on earth does this mean? How are they not mice? It's not that they don't have whiskers that makes the difference, it's that their genomes have different buffering capability for different point mutations. And what I'm arguing here is that understanding this buffering capability will give us new insights about both diagnostics, but also about drug discovery as well. Here's the biggest discrepancy of it all, and I'm very happy because this actually comes back to a mutation that I do believe was discovered in a family from the city of Patras. Of course, this is Parkinson's disease, this is alpha-synuclein, and the famous A53T mutation, one of the very large pedigrees that first put it on. Well, here's a funny thing. A53T, but the mouse genome encodes 53T. Bah, people said. 53T, Parkinson, it doesn't matter. Mice don't live long enough. Yeah, that's fine, except that if you take the human alpha-synuclein protein encoding 53T and you make a transgene in the mouse, by age 1, that mouse will have lost all its dopaminergic neurons and will die. So mice do lose dopaminergic neurons. Bah, will say additional skeptics, it's because the human transgene was expressed too hard and this and that. Well, it turns out, of course it had to be mice and cats, didn't it? But, and it turns out the cat genome also has A53T. So we now have mice and cats encoding a mutation that has incredibly high liability for Parkinson's disease in humans, and neither mice nor cats get Parkinson's disease, as far as we know. Why is that? Well, to cut a very long story short, because I am mindful of the time, we have a situation in which there's a pair of amino acids, this one and this one, in the central region of alpha-synuclein that will protect from the aggregation, the pro-aggregation properties of the 53T mutation at the N-terminus. And the hypothesis is you have a pro-aggregation versus anti-aggregation tug of war going on. We don't know if this is completely true, but we are actually prosecuting this target in Galatea right now because we think that if we can make alpha-synuclein to fold a little bit more like it should with these two residues, we think this will be resistant to PD. We think that will be a really good thing. So now we know nothing about alpha-synuclein, stability, interactors, and so on and so forth. All right. So I'm going to switch gears now and talk about a little bit about de novo drug discovery. So, and the use of genetic suppressors. So the ciliopathies, many of you will have heard, is an umbrella group of about 100 disorders. I've been studying these disorders for the past 25 years. They are associated with a great many pathologies with the concomitant appreciation that the primary cilium is not only associated with disease of the kidney, and the reproductive tract, and the circulation of cerebrospinal fluid, but it's pretty much present in every cell type. In fact, you'll be hard-pressed to find a cell type that does not have the cilium. Okay, so we discovered many years ago that several of the proteins that involve the ciliopathies, most notably the proteins mutated in Bardet-Biedl syndrome, regulate taking out the trash. They regulate the function of the proteasome in degrading target proteins, particularly when it came to effector proteins, such as components regulating Wnt signaling. So in this particular review, and here's the original paper, we were able to show, and this has been replicated now many, many times, that if you suppress the function of a ciliopathy protein, you reduce the activity of things like Wnt and Sonic and Notch and NF-kappa beta and I-kappa and so on and so forth. Pretty much any paracrine pathway that operates selectively through proteolytic degradation is affected by ciliary dysfunction. And for this, we built this very nice assay. We didn't invent this assay. This is known in the field. This is called the super top flash assay. And it's an assay that is based on a very simple principle. If you have beta-catenin in abundance, beta-catenin will enter the nucleus, bind these TCF-LEF transcription binding sites, and initiate transcription. So if you have a cell line that has many of these TCF sites driving luciferase, then the more beta-catenin you have going in, the more luciferase you're going to produce. So we made cell lines that were stably doing this. We suppressed ciliary genes. Please note here that we have the rescue. As far as I'm concerned, unless it's a rescue experiment, I don't believe it. Sorry, I'm a little bit of a stickler to this. But then you get too much beta-cat, too much beta-cat, too much beta-cat rescue as a phenotype, and this was the phenotype. And the experimental design here was that we made these cells, these are retinal pigment epithelium cells, and we plated them on 148 384 well plates. Each one of them had a library of RNA, of siRNAs, that were representing the entire genome at about 5 siRNAs in duplicate per human gene. We stimulated the cells with wind after 72 hours. 4 hours later, we harvested, looked for luciferase activity, and also if they were alive, and then that was the end of the experiment. And the result was looking very stringent z-scores of -3 and replicated. We ended up with 29 genes. When we did secondary validation for axin 2 expression, which is a direct target of beta-catenin, we ended up with 13 genes. This is pretty good. That meant that the original screen was pretty decent because we retained almost 50% of the primary hits. And then the third thing is we did a genome harvesting. In other words, are any of these genes, so remember what we're asking for here. We're asking that our cells are missing a ciliopathy protein, and we're systematically suppressing every single gene in the genome, and we're trying to ask the question, are there any genes that if we suppress them, we rescue the overall phenotype? So it's a double mutant. 1 + 1 = 0. Now it's beginning to come in, right? There you go. So we end up with 6 genes that, when suppressed, rescued ciliary function, 1, and 2, had never been reported in humans to be associated with a loss of function disease. Because if the gene, when suppressed, is associated with another disorder, it doesn't make for an effective target. And this is cool. So the first gene that we studied was a gene called USP38. And this is the beauty of agnostic screens. You don't know what you're going to get. Well, it turns out that this is a ubiquitinase, a D-ubiquitinase, which makes perfect sense. But we know very little about this gene. And here's a nice result. I love seeing that result. This is a double suppression result in zebrafish embryos. So this is the wild type. This is the phenotype. This is convergence and extension. This is an early Wnt phenotype. And when we suppress BBS4, this is the phenotype that you see here. And USP38 by themselves, you see a little bit of pathology, not much. When you put them together, you have rescues. Not a perfect rescue, but darn good. Now, convergence and extension is not a great phenotype to study a human being because it's a gastrulation movement that happens in the first trimester. It's not a very useful indication, it's just a surrogate. So, however, retinal degeneration is something that happens very commonly in the ciliopathies and a major pathology we're interested in going after. So this we can study differently. So here, you're looking at cross-sections of the photoreceptor, and we have stained those for rhodopsin, because rhodopsin should all be in the outer segment of photoreceptors and not in the inner segment of photoreceptors. And as you can see here, we actually have remnants of rhodopsin we have quantified here. The other thing we can do, and I'm not going to belabor the point, is we can actually expose the animals to a functional test. Basically, you strobe light to these animals and they freak out. But if they can't see, they won't freak out. So if you strobe the animals and they don't move around, that means that they can't see. Fantastic, right? We published this in a nice journal. We were excited. We had a great target. And then what happened? Turns out that USP35 and its there's 2 proteins, 35 and 38, are terrible drug targets. They're globular, We cannot differentiate them very well structurally from other deubiquitinases. We showed these to, chemical biologists. We went to GSK, we went to other people, and everyone's like, no, you can't do this. I'm so sorry. This is after 6 years of work. So we had a few choices that we considered. And then we thought, hang on a minute. Can we snatch the victory from the jaws of defeat, as it were? What did we learn? USP35 or USP38 might not be a good targets, but we learned that if we augment the proteasome, we might be able to achieve rescue. So David Brun, a very talented scientist inside one of the two companies that we're running, Recindo Diagnostics, he developed this cell-based assay where essentially cells shine green when the proteasome is not working. The way he does this is that he took green fluorescent protein and he tagged it with a degron that is recognized by the proteasome. So, in a healthy cell, the cell sees the degron, tells the proteasome to go kill the GFP. But if the proteasome is not working very well, then the GFP will persist and you will identify green cells. And this is amenable to robotics and high throughput screening. This is a proof of concept expression where with DMSO you actually see a lot of green, but if you put one particular truck here, you get a rescue. I wouldn't recommend this drug. It's actually a pretty potent antifungal that those of you who wear contact lenses will have it in them. The little problem is that if you ingest it, it will burn your throat, your esophagus, then your stomach, and then you'll die. So it might not be the greatest drug. However, we were able to do a focus screen, and the way we did the screen is we took 5,000 compounds. Most of them were FDA approved. We did an in vitro screen, and then we took all the positives from the in vitro screen, and we tested them in the in vivo model. And to cut a very long story short, actually not that long a story, but it felt like a long story, we identified 2 strong leads and 4 backup. And this is the key experiment, as far as I'm concerned, with compounds C11 and C27. This is the animal. You acclimate it, and then you take it through light and dark transitions. This is a zebrafish embryo about 5 days later, and these are CRISPR mutants now. And you expose them to the drug about day 1, daily washouts, and then more. And this is the number of responses that you get. So if you're wild-type animals, you get about 3.5 responses during the experiment. If you're a mutant, the CRISPR, you get only about 1 response per light-dark transitions. And when we expose these animals to the light, to the drug C1127, we got complete rescue. And this was concomitant with the restoration of the distribution of rhodopsin to the outer segment. And I really think we have a drug. We're moving hard on this. We're doing PK/PD in the mouse, efficacy testing. And we actually have an access to a BBS macaque model, which is very useful. So we're really planning for the future. All right. So my last little vignette, and I really want to talk about surprising gene discovery. So, and to reinforce the point that agnostic discovery has intrinsic value. So many years ago, colleagues of mine from Cologne contacted me because Bernd Volnick said to me, I just saw this young man. He's a classical Kabuki syndrome boy. He presents with all the clinical features. He does not have the known Kabuki syndrome mutations, but he had what looked like a pathogenic mutation in RAP1A through uniparental isodisomy. He had inherited 2 big chunks of chromosome 1 from dad. And that had converted this mutation here, the arginine to threonine, into homozygosity. So of course, I'm not going to show you all the data. We actually made a zebrafish model for this, and we're able to show indeed that this pathogenic mutation was this mutation was pathogenic, and it was recapitulating many of the key features of Kabuki syndrome. But this is where it gets really interesting. So Kabuki syndrome, all the other genes mutating Kabuki syndrome are methyltransferases. And everybody's thinking about methylation regulation, epigenetic phenomena, da, da, da. If you think about this, I'm sorry, I don't buy it. Because if you have global methylation phenotypes, you're not going to have Kabuki syndrome, you're going to be dead. So what is it that gives specificity to Kabuki syndrome? I do not know. So one hypothesis is that we know what RAF1A does. It either regulates BRAF or RAF1. And this is a cascade involved with a group of disorders that many of us know, of course, the rasopathies. Noonan, Costello, CFC, Leopard, and others. And in fact, if you look very carefully inside the pathophysiology of Kabuki syndrome, you actually observe a clinical overlap with these disorders. Not a perfect overlap, but there's a distinct overlap. So we went ahead to study this because we said, well, which of these activities of RAF1A? Is it BRAF or is it RAF1? And it turns out it's RAF1. And here's the key experiment, and I'm going back to 1 + 1 = 0. This is a zebrafish. These are zebrafish embryos. We can biochemically measure how much phosphorylated MEC we have compared to total MEC, and we can actually see the phenotype. But here's the key result. We argued that if this is true, if we genetically suppress RAF1 in our Kabuki model, we should see rescue. I really want to go back to here, right? Because RAF1 is an inhibitor of RAF1. Therefore, if I don't have RAF1, I should have excessive RAF1, I should have excessive MEC signaling. Therefore, if I suppress this guy, I should attain some degree of homeostasis. Make sense? I know it's these pathways, they always give me a headache. And I have spent years looking at this, so I now expect you all to absorb this in 3 minutes. But we'll get through it. So this is the experiment. This is again, percentage of embryos with early gastrulation defects. This is an easy thing to look at. This is the jaw length. So all children, or almost all children, with Kabuki syndrome will be hypognathic. And we can actually measure the relative length of the upper jaw versus the lower jaw in zebrafish embryos. And it turned out this is the case. And it turned out that if we suppress RAF1, we saw partial rescue of early developmental defects and almost complete rescue, not complete rescue, arguing that this is not a complete rescue, maybe not be the whole story of the phenotype. Nonetheless, based on this data, we said, hang on. For RAF1 and derivative products, there's a ton of small molecules out there because all these genes are heavily involved in cancer. So we took 29 known RAS inhibitors against melanoma, against colorectal cancer, against various things that are FDA approved for these uses, and we tested them for conversion extension and jaw defects. And we identified 2 that completely rescue the phenotype. These are safe drugs. In fact, one of them is dabrafenib, which is a very famous drug for melanoma, for the V600E, activating mutations in melanoma. And once again, early gastrulation movements, I don't care what it looks like. It is not very helpful. And also, correcting the jaw defects, I really don't care if a kiddo has a long jaw or a short jaw. I just want him and her to have a full and productive life. So we looked at neurogenesis. And we were able to rescue neurogenesis in mutants. And once again, this is the total numbers. This is now not just RAP1A, but this is various different genes, each one of which has been associated with Kabuki syndrome. And in each case, we were able to observe significant rescue of the total number of neurons born during neuronal development in the fish. And last but not least, we talked to Bill Wetzel, who's been running pharmacology experiments at Duke for more years than I can think of, and in collaboration with his manager Ramona, they actually were able to devise a test in the mouse. I can't show you. I can't show you all the data, but the brief summary is this. The key experiment here is that we treated mice postnatally, 2 weeks postnatally, and then we tested for efficacy 4 weeks after that. So this is the window that we'll be looking at kids. So children born with Kabuki syndrome are usually diagnosed within the first few months or year and a half of life. And here we're looking at spatial awareness and also memory. You have these fun little objects and you measure the length of time that the mice spend with new objects and how long it takes them. And what we found, and this is a year and a half, very expensive and very tedious work in one little sentence, but again, this is what science is. Postnatal, between P14 and P21 administration of dabrafenib, at 50% of the human dose. So half effective human dose, completely rescued neurodevelopmental phenotypes as assessed at P30, P40. So based on this now, we've moved full tilt to humans. We got FDA awarded us a rare disease designation for dabrafenib in October 2020, and an SPIR started coming here, a partnership here, and then around here, something called COVID showed up, and everything froze. But we are optimistic that we will see the other end of this horrible calamity for humanity and move forward with this. So I'm going to close. I was a young postdoc, starry-eyed postdoc at Baylor College of Medicine when this paper was published on the 21st of April, anniversary of DNA Day, anniversary of the first paper. This is, of course, the public effort. There was another paper in Science. But what I remember with great clarity is that back in the day, my leaders promised me that she's going to feel better. It's my daughter, actually, and she's feeling fine, thank you. But there's many other girls who are not. But I had many of our leaders promise me that because of the genome and the $3 billion that was spent, we're going to do like, I remember how cancer was going to be obliterated by 2015. That didn't quite work out. So I'm going to say that there is no question that the completion of the genome and the effort that everybody, including people in this room, are putting in is improving and changing people's lives. However, I must caution my colleagues, with the warmest intent that I have, that we have been over-promising and under-delivering on this for a very long time. And it's time we get our together, or else, especially at a time where trust in science is eroding because of the influence of social media, unless we get our together, we're going to lose the confidence of the public. We have made exceptional promises about healthcare delivery and genomic medicine and precision medicine. I will go on record saying I deeply admire the work and the progress that has been made, and I congratulate my colleagues who have done this. However, we have a long way to go to get there, and I really hope I'm still around to do this, because I want this guy, that's my youngest son, to actually feel better. Well, and I'll stop there. Thank you very much for your attention. Τα καταφέραμε.
So, Nico, thank you so much. That was a really inspiring talk. I think you touched upon things that we were hardly anticipating. In principle, you demonstrated that what we knew as a balancing effect of different variants, then you see the same effect on the very same gene. So we knew the modifier genes, which is basically a defect in a different gene that alleviates what happens in another one. But what you've shown us here today is that you can have the same variant in the same gene that can cause the same
That's exactly
effect.
right.
Yeah.
In fact, what is interesting, I'm a big fan of modifiers everywhere. Oh, and by the way, I can tell you an anecdote. It was the early days of the ciliopathy, and I was full of beans, all the hot papers were coming out. I was like, oh, I'm the man. And then I'm giving a chastening talk at the University of California, San Diego, and a very, very senior person comes up to me, whom I respect, and says, I don't get it. What's the big deal? We've known about this since the 1950s. And it is true that modifiers and modification and all this stuff is established in cell biology and in animal models for a very, very long time. I mean, in Kansas, there's been a breeding of chickens going on for the last 90 years based on these paradigms, right? And I had to sort of chasten myself and say, yeah, but those of us doing human genetics haven't quite read all the literature. But it's true, I always thought about trans effects, as you described one into the other. But it turns out that cis effects might actually have a greater potency. I don't know why. My hypothesis is that it is because they have a higher ability to impart a change in protein function. So it's easier to change the function of the protein from within than from another gene next door. Now, the second question I always get is, can you get this interaction phenomena, epistasis, in trans? And my answer to you is, I wish, but because the space becomes all the allele combination squared, because you have two, and then to the power of 3 to the power of 4, it becomes very difficult. The one exception is surfaces of receptors with ligands. So you can take the surface of a ligand and surface of a receptor, and you can think about protein-protein interaction, where you can imagine that a modification here and a matching modification there will have a compensatory mechanism. But that's theoretical. I haven't figured this out yet.
Right. And we can all think, of course, of the pharmaceutical potential, the therapeutic potential.
Yes, because what I'm thinking about is that the computational modelers are getting scary good at precisely modeling the docking of small molecules onto proteins. What I'm thinking is, if we can understand the change in shape of a protein with a compensatory mutation, can we mimic that change in shape with a small molecule? That's the idea. Yeah. That's still science fiction, but if it turns out to be true, I think it's going to accelerate drug discovery.
Well, we all need to find out.
We will find out.
Any questions from the audience?
Oh, come on, don't be shy.
English, Greek, Spanish? None?
It's okay. All right.
That's fine. So, thanks again for being here with us. Yeah, it's been a pleasure. Inspiring talk as always, right?
Thank you everybody for your attention, and good luck with your work.
Λοιπόν, στην πραγματικότητα αυτό ολοκληρώνει το κύριο μέρος του συνεδρίου. Αύριο έχουμε το φεστιβάλ της γενετικής της εξατομικευμένης ιατρικής, το οποίο περιλαμβάνει μία θεατρική παράσταση, DNA Look Who's Talking Now, και νομίζω είναι πάρα πολύ ενδιαφέρουσα στο ό,τι αφορά το περιεχόμενο. Εγώ τουλάχιστον εξεπλάγην τόσο θετικά την πρώτη φορά που την είδα, που νομίζω ότι αξίζει τον κόπο έστω και το πρωινό ξύπνημα ή τον έρθε κατευθείαν το μπαράκι. Θα έχει μετά μία στρογγυλή τράπεζα που θα περιλαμβάνει ομιλητές σχετικά με θέματα που αφορούν τη γενετική, και θα είναι π.χ. RNA εμβόλια, υγρή βιοψία και μία συζήτηση μεταξύ τους. Και μετά θα έχουμε δύο παράλληλα events. Το ένα θα είναι μία έκθεση σε μορφή DIY, do it yourself, η οποία έρχεται από το Μουσείο Smithsonian της Νέας Υόρκης, της Washington, με συγχωρείτε, που περιλαμβάνει θέματα που άπτονται της γενετικής εξατομικευμένης ιατρικής, Unlocking Life's Code, αυτός είναι ο τίτλος της έκθεσης, η οποία ξεκίνησε το 2014, διαζώσεις στο Μουσείο Smithsonian της Washington, και έκτοτε ταξιδεύει σε όλο τον κόσμο τη μορφή do it yourself. Παράλληλα, θα υπάρχει επίδειξη του φορητού εργαστηρίου μοριακής βιολογίας, το οποίο τρέχει από το εργαστήριό μας εδώ και αρκετά χρόνια, με πολύ σημαντικά παραδοτέα και στην εκπαίδευση αλλά και στη διάγνωση, το οποίο μάλιστα ήταν και μέρος του project iGEM Πάτρας 2020 που προβέθηκε με τα σημαίνιο μετάλλιο. Οπότε πιστεύω ότι η αυριανή μέρα θα είναι πραγματικά ένα πολύ σημαντικό happening, το οποίο θα συνεχίσει και τα επόμενα χρόνια. Οπότε σας περιμένουμε αύριο από τις 8.30 και μετά μέχρι τις 5 περίπου, 5-5.30 στο χώρο το συγκεκριμένο. Λοιπόν, ευχαριστώ όλους για την συμμετοχή σας σήμερα και καλό απόγευμα, θα σας δούμε αύριο.